We are live.
Okay. Can I start?
Just a second. Yeah.
Okay. Distinguished speaker of the day, Dr. Gitanjali Yadav, colleagues and students from Shastra University and other institutes, a very good afternoon to all. On behalf of the Shastra fraternity, it is a great privilege to welcome you all to the Shastra National Science Day Award lecture for the year 2021 by Dr. Gitanjali Yadav. The future belongs to the curious, says a famous quote. It is a pleasure to introduce Dr. Gitanjali Yadav, who is an exemplar of this quote. The ability of plants to synthesize their own food is a fascinating phenomena for all kids who study photosynthesis in their school days. I distinctly remember how I wish to have 2 leaves on the top of my head so that I can escape my mom's scolds for not eating properly. I'm sure every one of us have had such dreams and fantasies. However, least did we realize then that this process of photosynthesis, catalyzed by an enzyme called rubisco, is one of the most inefficient phenomena in the world. However, plants have evolved mechanisms to overcome this inefficiency through various means. What are these mechanisms? And can we harness these for better crop productivity to address food security issues of the world? This curiosity, coupled with social commitment, is what has been driving Dr. Gitanjali Yadav for the past few decades. Her career trajectory is an inspiration for us. A botanist by training from Sri Venkateshwara College, University of Delhi, she moved on to biomedical research for her MSc and then did her PhD in bioinformatics from the National Institute of Immunology, New Delhi, where she worked on polyploidy synthesis. With this vast experience that she had gained, she returned back to plant research, combining several diverse disciplines and techniques, including omics approaches, data science, artificial intelligence, etc. Currently, she is a scientist at the National Institute of Plant Genome Research. She is also a lecturer at the Department of Plant Sciences, University of Cambridge. Ma'am, your career trajectory and research is a telling example of how young budding scientists should shed our engrossment with a single technique and shift focus to address biological questions using multidisciplinary approaches. Dr. Gitanjali Yadav is also active in science communication and outreach. She does a lot of outreach activities in India and Cambridge with a specific focus on women empowerment in science. She is the proud winner of the Sastra Obaid Sidiki Award 2021. With no further ado, I hand it over to Dr. Gitanjali Yadav to deliver the Shastra Obaid Sidiki Award lecture. Ma'am.
Thank you for that pleasant introduction. And good evening, everyone who has tuned in to the National Science Day lectures today. As many of you may be aware already, the National Science Day marks the discovery of the Raman effect by Indian physicist Sir C.V. Raman, Bharat Ratna, and he discovered this effect in 1928. If you all have, for those of you who attended Professor Ghatak's Sastra Award lecture this morning, about Raman effect and the scattering of light and the interesting stuff that was being done in physics at that time in India, leading to the Nobel Prize that Sir C.V. Raman got in the year 1930. So those of you who missed Professor Ghatak's presentation, I think you're more than welcome to go back to the Sastra website and be able to, it was live streamed and I'm sure it is available currently and you will be able to visualize that. I will pick up the thread of the story from the point of the Nobel Prize. So in 1930, the Nobel Prize was given to Sir C.V. Raman for the Raman effect indeed. But 2 years later, in 1932, far away in the Basti district of Uttar Pradesh, there was born a child, a boy who studied botany in the early years of his life, but went on to become a sparkling and globally renowned neurogeneticist, after whom this Life Science Award of Sastra has been named for the National Science Day. Professor Obaid Sidiki And he was invited by Sir Homi Bhabha to set up a laboratory at the Tata Institute of Fundamental Research, Bombay, TIFA, in 1962. 30 years after that day, he became the founding member of and the director of TIFA NCBS, where he met 23 years ago, he met a group of students, visitors who had come just for that day to see the laboratories. And one of them was a half confused, half happy, half overdosed on adrenaline girl. That girl was I. I guess what I'm trying to say is that with this preamble is that you don't need to have deep philosophical reflections or introspective, meditations, or even a degree in an Ivy League college to decide what you want to do with your life. All it takes sometimes is just one conversation with one person, and it can happen when you're least expecting it, and they can change the course of your life. So that was 23 years ago when I met the man who Shahid Jamil appropriately calls the gentle genius of biology in India, Padma Bhushan, Padma Vibhushan, FRS, Professor Ubaid Siddiqui, whose picture you see on the slide right now, and whose seminal work established how taste and smell are detected and encoded in the brain. The one thing about him that was unmatched then, and that remains unmatched even today, is how he treated me, us, all youngsters, as if we were geniuses. He insisted that we address him as Ubaid, first name basis from the first moment onwards. At that time, I didn't know anything about Drosophila. That was the species he was working on, but he didn't let that become a handicap for me. In fact, we spoke about plants. And he too had been a botanist, you see, as I said, just like me. And so that's how he connected with everybody. And he made you feel comfortable with yourself about who you were and what you wanted to do. I went to TIFA, not sure about what I wanted to do next in life. That was that stage of life where each one of us, I'm sure each one of us know what it is to have a stem cell stage in your life, when you have it in you to become anything and anyone, because you're open to ideas. I was then. And I would have been equally happy at that time if I had taken up mass communication or quantum physics or administration, business management, even professional swimming for a career. But I made Ubaid, and Ubaid crystallized the future for me, at least. I came back from TIFA with the conviction that I wanted to pursue scientific research as a career. And it was such a relief to finally, know who you wanted to become. I always wanted to become who I wanted to become. And so he spoke to us about plants. A single plant, and that is what I'm going to talk about today. Can you see my slides?
Yes, ma'am.
Great. So this is my story from what conversations we had with Ubaid. And a single plant, as he said, can exhibit many forms. Its ability to do so is its ticket to survival. If one form is susceptible to a particular disease or an environmental change, for example, another form may not be. So your chances of surviving and prospering as a species are greatly enhanced if you look at the world with many, faces. In the case of plants, a particularly successful way of doing this, meaning looking at the world with many faces, is through gaining an additional set or sets of genetic material. This extraordinary phenomenon, it may also hold the clue to one of the greatest botanical mysteries, also called Darwin's abominable mystery. That is, why did flowering plants evolve so rapidly around 130 million years ago? So it was the first time this was noted was by German botanist Hugo de Vries, who in 1903 was the first to write about the many faces of flowering plants. Hugo de Vries, for those of you who may remember, he was one of the three scientists who had recently rediscovered the work of Gregor Johann Mendel. And as a result, he was at that time beginning to question Darwin's theories of evolution. De Vries became convinced that species must at some periods have evolved in sudden bursts, the so-called discontinuous variation, rather than Darwin's theory of continuous slow accumulation of genetic traits. He found, De Vries found, he got evidence at that time in an abandoned potato field where there used to be this, which he used to be walking past in the evenings, and there was this cluster of evening primroses that had established itself in this field. You see primroses on the slide just now. De Vries noticed that among these evening primroses, there were new types, and they were only subtly different from the parental types, but they were definitely distinct. What was interesting about them, about these distinct ones, the bigger primroses, was that they didn't interbreed with the olden types, with the old types. So they remained stable from day one, from the day he noted that there's a bigger flower or a bigger plant, they remained stable. And this is what Obaid first talked to me about. He also was very interested in chromosomes and mutations. So De Vries then, when he noted these primroses, he talks about the fulfillment of all his hopes when he found these plants growing. There were no steps, there were no intermediate generations, there was no struggle for existence, so to speak, in the classical Darwinian sense. These plants just seemed to grow overnight. And one of them, which he called Unithera lamarckiana gigas, that was the giant one. So researchers eventually figured out what was happening there, and they discovered that the reason this plant was so big was that the chromosomes of the plant had been duplicated, the entire genetic material duplicated. So for those of you who are new to this, normally, of course, in a plant, the pollen and the ovule, they both have a single set of chromosomes, and they're called haploid. When they fuse, they form a zygote, and that is the beginning of a new plant. And this new plant, the zygote, ends up with two sets of chromosomes, okay? And that's called diploid, one set from mom, one set from dad. And so each cell in the plant then has two sets, one from each parent essentially. But these ones, the primroses, the gigas essentially, had 3, even 4 sets of chromosomes. So quite literally, the amount of genetic material in the plant cell nucleus had multiplied by a factor of 3 or 4. It's a curious phenomenon, the one that I've just talked to you about, and it's earned itself a curious name. It's called polyploidy. And the word itself means many forms. And this happens when something goes wrong in the process of cell division. This results in an additional set of genetic material. In practice, this means that within one generation, a new species can indeed develop. So you're seeing a complete distraction from what Darwin had said, natural selection over millions of years of evolution. In animals, this kind of an abnormality of cell division, the one I just spoke about, polyploidy, it is extremely rare. You might see in some lizards, some fish, but very rare. In plants, polyploidy is rampant. It is in fact estimated that half to 2/3 of all flowering plants are polyploid, including more than 99% of ferns, as well as 80% species of the grass family. And the grass family, for those of you who don't know, the grass family is the family that is the source of all your important crops, the cereals, rice, wheat, barley, oats, corn. So similarly, so crops are polyploid, and that's one major reason for their success. Similarly, just like crops, the grass crops, there's also a huge proportion of our other crops, including sugarcane, potatoes, sweet potatoes, bananas, strawberries, apples. So all of these are essentially polyploid, and there's a lot of work. For those of you who are interested, I would love for you to take a look at a couple of papers, or maybe even there are lots of good YouTube videos on these flowers of the phlox and petunia, on which a lot of polyploidy research has happened. Pansies also. There's so many types of petunias and so many types of phlox and so many types of pansies that you will find around you. These are there because of polyploidy. And there's a lot of new technologies that are helping us to understand polyploidy, identify polyploidy. See, the thing is that you can't really just look at a plant and say what its ploidy status will be. So the way to do it is to be able to look at the number of, the nuclei and the chromosomes within that. And this is done by flow cytometry. There are many other ways also to look at ploidy and the level of ploidy, but the incredible part is that plants somehow are able to inform their pollinators, these insects that are fertilizing them, pollinating them, dispersing their seeds, are able to inform or tell their pollinators. Somehow these insects are able to, just by looking at the plant, they can say whether it's a tetraploid or a diploid. There is just no way we can do that, but plants can do, but insects can do that. And that is another incredible part of nature and the plant insect interaction. So, as I said, there's rapid advances that have happened and taken place in technologies like flow cytometry, also next generation sequencing that we are doing a lot of. And these could help address, as I said, back to one of evolution's greatest mysteries. So what is that greatest mystery? Again, why so soon after appearing in the fossil record about 130 million years ago, did flowering plants or higher plants suddenly explode into the bewildering diversity of species with their equally bewildering range of reproductive techniques? Showy magnolias, you have multi-petal blossoms fertilized by bees, you have plants that cast their pollen to the winds, and so on. Over 70 species of plants, almost all the flowering plants, I would say all the families of flowering plants that we have around us, diversified during this event in time. And it is so difficult to actually find out what happened such a long time ago in the past. It may even, it may be that we can tie that burst of diversification to a polyploidy event. And there is some amount of circumstantial evidence that a round of polyploidy did occur in the major plant lineages, at least, because in today's genomes, in today's present day plants, we can see remnants of that past in the present genomes. And that's some of the work that I do in my group in India as well as back in the UK, because there are these extra multiple copies of the controlling sequences. We sometimes call them gene families, and they could well be the smoking gun or the signatures of past polyploidy events. They are currently retained in the present day plants as even though much of the DNA has been completely replaced from that time. And it was just about 10 million years during that time, 130 million years ago. And that's why, because of that little window, it's so difficult for the fossil record to really give you something substantial or to let you see how to dissect out that smaller part of the larger history of flowering, of evolution of flowers and flowering plants, or angiosperms as we call them. So what we think is that polyploidy is truly very likely one of the reasons why angiosperms, first of all, were successful, but secondly, the reason why they are so productive, that we can truly feed 8 billion people of Earth on their back. As I said already, many of our crop species, wheat, maize, sugarcane, you name it, they are polyploid. And just last month, 2-3 weeks ago, there was this paper published in Nature, Ecology and Evolution that suggested that potentially flowering plants may be 100 million years even older than what we thought. So imagine how difficult this is to work out, because you're still trying to work out what happened in those 10 million years, 130 million years ago. And then suddenly somebody says, you've got to go back 230 million years ago and then figure out what happens. But truly, what we believe is what happened was polyploidy, and the signatures of that event are currently visible in the transcription factors or regulators. I don't want to use terms like transcription factors without giving a preamble about it, but you can see them in sequences, in pieces of sequences in present day genomes, because we've been looking at and comparing these genomes all the time, as I will talk about when I come to the work that I do. So there aren't many studies yet, but understanding this curious and strange phenomenon, polyploidy, is something that may hold the key to the evolution of flowering plants. Not just evolution of flowering plants, also the understanding of plant-insect interaction in ecosystem. You see, there are knock-on effects of everything. And so I told you there are insects that can recognize ploidy, even though no technology, no human, no mechanism, and no amount of artificial intelligence, we haven't applied it yet. I'm trying very hard, at least in India, to start out AI for plants, and we would be able to have enough data to try and do some kind of learning to be able to predict from, but it's still very difficult, altogether not known, to look at how, what is the ploidy status of this. But anyway, understanding ploidy better in the future, as I said, may hold the keys to the evolution of flowering plants. Also, their ecosystem level interactions, and also the quality plus quantity of some of our most stable food sources. I will talk about, this is not just simply a matter of pure research. It's sometimes, by the way I speak, I think it just suggests, it looks like it's a matter of pure research, but its practical implications are all around us. And as I will talk about now in my current research, we think that the practical implications may indeed provide resilience to future climate and disease outbreaks, which brings me to one of the biggest climate crises that the world or the planet is facing right now, which is why I am not in front of you today to give you this lecture physically, be able to look at your faces, but here I am staring at a computer, not sure if the Zoom connection is still up or not, but I will continue to speak till somebody tells me that something's gone wrong. So, we are online, we are all remote, we are separated. And this is a picture that I took in the Lake District 2 years, 3 years ago. And each day for each one of us, I think the current COVID pandemic basically just reminds you, the value of the great outdoors, bringing home the necessity of working together for a better future. There is no alternative. We've got to work together for preserving Earth's natural resources and address global challenges, urbanization, deforestation, finding ways to feed an increasing population, fuel security, food security, and above all, climate change. As many of you may be aware, much of climate change, in fact, my 8-year-old son is aware that much of climate change is driven by the rising levels of atmospheric carbon dioxide. These levels, which are currently at about roughly 440 parts per million volume, these levels are higher than what the Earth has seen in the last 800,000 years. Okay, so it is not true that the current COVID lockdown basically has brought, all right, it is true. It is true that the COVID lockdown did bring down much of these emissions to a halt for the time being, but we are beginning to open up again. And who's to say that the world or we would not be speeding back into our jet planes the moment we get a chance? And so I do hope that some of us who are listening to me, and I do hope some students are listening to me, and they are able to reflect upon the carbon footprint that we are leaving on this planet and how it can take tens of thousands of years, for all that emitted carbon to make its way back into the Earth's crust. Leaving aside the causes of this increase in atmospheric carbon levels, that's a different story for a different lecture and a different series of talks altogether. What we need to do is we need to find ways of bringing this level of carbon down. And once again, this is where plants come into the rescue. Okay, so I'm Gitanjali Yadav, and I'm a plant scientist. I work at the Department of Plant Sciences, Cambridge University, and I also hold a joint appointment, as was presented in my nice introduction today. My joint appointment is at the National Institute of Plant Genome Research, New Delhi. Today, I will be talking about green carbon for food security. Even though many of us are aware that plants are the natural air filters of the earth, we still fail to appreciate the true value of the natural capital of plants and of photosynthesis, that single process by which plants are able to A, draw down atmospheric carbon, B, fix the inorganic carbon into organic carbon, and the carbon that all life forms are made up of, otherwise known as food, and C, release pure oxygen back into the air, cleaning it up. As researchers, the question that we are asking are two questions. One, what are the plants that are most efficient at photosynthesis? And two, where do they live? And most importantly, of course, can we understand them better to improve other less efficient plants? Surprisingly, the answer to the first question is in a tiny unicellular green alga that lives all around us, mostly in freshwater habitats, but also soil environments, the oceans, snow, you name it, and it is there. Okay, so there's also a lot of ocean phytoplankton that make up less than 1% of the total biomass of plants, but they are responsible for nearly half the net photosynthesis on Earth. So research has shown that green algae are able to fix carbon up to 40 times better than higher plants. So what is it in green alga that makes them do photosynthesis so much better? To understand how they're able to do this, you've got to first understand the issue of efficiency altogether in photosynthesis. It was explained a little bit when my introduction was being given at the beginning of this talk, but to make you understand better, the key to understanding photosynthesis and the efficiency and the low and high efficiency of different species is the manner in which green plants have managed to rectify a flaw that is at the heart of photosynthesis. The flaw is this. So Rubisco, the global carbon fixing enzyme, the guy that stands at the interface of living and non-living, this guy, Rubisco, performs less well in most plants. And it performs less well due to its inability to distinguish atmospheric carbon dioxide from atmospheric oxygen. Now you see there is so much in the air, there's 21%, there's so much oxygen compared to 0.04 of carbon dioxide. So Rubisco will, when it sees the atmosphere, it sees more oxygen straight away, and its active site is such that it cannot distinguish between the two. So it takes up oxygen instead of carbon dioxide. So basically what's happening is a wrong number being dialed by making huge amounts of, so how do plants overcome this wrong number? By taking, that Rubisco should not take oxygen instead of carbon dioxide. What plants do is that they make huge amounts of Rubisco, so much so that Rubisco is the most abundant protein on this planet. And yet it is not enough. How would it be when oxygen levels are clearly, as I said, much higher than the levels of carbon dioxide in the air? And so back to the question, how do algae fix this flaw? And is there something we can learn from alga that can tell us how we might be able to fix it ourselves? With that, I would like to introduce you to the model unicellular green alga that we use in the lab. And there it is. Hello. That fixes carbon very well. This little beast is called Chlamydomonas reinhardtii. It's a tiny little plant form that we are working with to understand the business of super efficient removal or fixation or sequestration of carbon from the atmosphere. The ingenious trick that Clammy, we call it Clammy, the ingenious trick that Clammy has evolved is that it physically separates Rubisco from oxygen. So the problem with Rubisco, the flaw with Rubisco is that it takes in oxygen as a wrong number. So just separate it physically. So what Clammy has done is that it packages Rubisco in a tiny little compartment called the pyrenoid. On this slide, you can see the pyrenoid, which is a purple colored little blob. It's a micro compartment, and it's called the pyrenoid, of course, as you can see in the figure. But the interesting thing about the pyrenoid is that it's chock full of Rubisco. So there is no Rubisco outside of the pyrenoid, and there is no way oxygen can enter the pyrenoid. So what is it that prevents oxygen from coming into the pyrenoid? There are all sorts of things. It's a concentration camp for carbon dioxide only. And there are proteins, there are transporters, there are all sorts of mechanisms that have evolved to prevent oxygen from getting in, and only carbon dioxide comes in. And so therefore, because Rubisco doesn't see the oxygen, it doesn't make mistakes. And because it doesn't make mistakes, it is extremely efficient. And such mechanisms that prevent physically or separate physically Rubisco from oxygen by concentrating all of the CO2 around Rubisco, these mechanisms are called carbon concentrating mechanisms, or CCM. And we are working on the biophysical CCM. There are three different kinds of CCM, of course, but we're working on the biophysical CCM in Clammy. There is blue-green alga, and there are also C4 plants and camp plants, which have evolved their own ways of concentrating carbon around Rubisco. And what we are trying to do in the lab, in our research, is to identify the key components of Clammy biophysical CCM in the hope of using it to improve crops, or to understand this whole process of the CCM and efficiency of photosynthesis. So this brings me to the work that we are doing with my collaborator and colleague, Professor Howard Griffiths at the Department of Plant Sciences. Our research is to interrogate very large genomic data sets. I told you at the beginning, we are looking at genomes, and we're looking at, we're trying to find molecular fingerprints or patterns in the DNA or the RNA that may help us in identifying finer details of the process of carbon concentration. In our case, it's algal carbon sequestration. And so what we've done is that we've collated over 50 important genes, which we feel are important based on several decades of published literature of the work that has been done in the past. And then we took these 50 genes, which we identified. We call them the core set of genes, or we call them the CLAMI core component of carbon concentration. So it's called the CCCCC5C. And then we divided the 50 genes into time. Which ones of those 50 are active in the morning? Which ones of those 50 are active at dusk, pre-dawn? So you can see now we have separated the gene expression data by time. So this is called a temporal resolution of gene expression. So essentially, this is on this slide. What you see is a heat map, and each row is one of the 50 genes that we're interested in, the core set of genes, and each column is one time point in the day-night cycle of growth of CLAMI. So in this picture, the green colors are essentially telling you which are the genes out of the 50 that light up at a certain time point, like the morning, pre-dawn, or dusk, or during the day when really photosynthesis is happening. Now, for each of these 5 time segments, so all of our data has been divided into 5 time segments. For each of the 5 time segments that you see in this heat map, we call them division, resting, pre-dawn, active CCM, and dusk. And you can see them on the 24-hour cycle up on the left. We have extracted the genes that get switched, the bright green colors that you see in the vertical box. So what we do is we extract those few bright green colored genes, and then we build gene correlation networks to understand how the genes in that time segment are associated with all the other genes of CLAMI. And when I say all the other genes, there are 17,000 genes in CLAMI. So we are looking at just 50 core, and out of the 50 core, during division, maybe 10, maybe 15, and then we relate them to the rest of the genome. So most of this work has been done by my PhD student, C2. And in this slide, you can visualize the CLAMI CCM gene correlation networks at every time segment that C2 has built. And what we observed, and as you can see in this figure, is that there are critical regulatory subnetworks that are active at different time points. Most importantly, we think that there is a unique set of regulatory genes that are controlling, the trigger of CCM, which are active only just before dawn. So if you look at the pre-dawn network, which is the second network, take a look at it, and you will find bright orange colored nodes. Each orange dot basically is a gene. So those bright orange colored nodes get switched on only during this time segment specifically. So we are now, so this activity, because it's taking place just a few hours before the onset of light, when the plant starts to see light and respond to it with photosynthesis, active CCM kicks in then. And we are now in a position, because of C2's work, to explore this little set of controllers or gene controllers in more detail and identify the missing links in the regulation of CLAMI CCM. So we've now decided to go ahead and do supervised and unsupervised machine learning on each one of these temporal networks in order to identify what these controllers are controlling. And in the long term, we hope that some of our findings from molecular research, in the long term, we hope that some of our findings from the molecular research may be translated to the field for higher plants. I think you heard my practice run on this one. And much of this work is funded by the Tigress program, a Global Challenge Research Grow Award for transforming India's green revolution by research and empowerment for sustainable food supplies. It is very encouraging for us to know that some of our collaborators at Princeton and York University, they've been able to successfully get some elements of the biophysical CCM into higher plants already, into tobacco. So we've been trying to replicate this in tobacco. And for me personally, the Tigress program has been an amazing roller coaster ride, encompassing much more than cutting edge research in green carbon. So Tigress, these are our colleagues and friends on the Tigress program. It's been a fabulous learning experience, as I said, bringing home the amazing benefits of working at the intersection of multiple fields and multiple technologies. So between 6 multidisciplinary, what do we call them, sub-projects or flagship projects, and over 30 organizations across India and the UK, various Tigris partners have joined hands across the disciplinary silos of natural sciences and social sciences. So in this program, we use high resolution land records from rural India. We use genomes, we use fossil records of pollen from ancient civilizations. 2 years back, there was this fantastic meeting before the pandemic. We all went to Rani Khedi together when the discovery was made that you have something bigger than Mohenjo-daro and Harappa. And we also use supply chain modeling. We use agriculture economics. We talk to farmers. We work with women's collectives. We use robots, geography, mathematical simulations, all kinds of data analytics to address the global challenge of feeding an ever increasing population. Most importantly in Tigris, and which is new for me because in my past 10 years of being a principal investigator, I had been doing only research in work. But after Tigris, I learned how important it is to connect your science to society. So most importantly in Tigris, we teach and train rural and urban youth to contribute to capacity building. There's a lot of this that we have done at the India-UK interface, but mostly by going back and talking to farmers, by talking to children in schools and asking them to connect with science. And I now find myself in a strange life comes full circle moment when I first met Obaid and he got me to choose science out of all the things in the world that were. And I think that even if one student who might listen to me sometime and make that choice of pursuing science instead of all that is there in the universe, I think that would be the most fantastic outcome of the outreach efforts that we are making. But what we've done more recently is not just rural outreach or getting farmers or lady farmers to learn new skills. There's also a lot of, so at Cambridge, I have been involved in bioinformatics, so training computing skills. We train students, not just students, but also entrepreneurs, business people, or working people, postdocs, even PIs, in the skills that are important for data science as of today. And I believe that in the future, there is just, if there is one word that can sum up all of biology, it's going to be integration. So you've got to know how to integrate your research and your science with all that is there out there in the universe. And the only way to be able to connect your data with somebody else's is to be able to know what they have done. And therefore, what you need to know is how to integrate your data with the existing knowledge base out there. And for that, you need to know, you need to have skills for computing. And this pandemic has just reiterated this even more so, because now all of us are dependent on communicating online via computers. So those of us who had not earlier used them are using them all the time now. People like me who were already using computers are even more, like I have 5 computers, if you can see me around me right now. So there's one for just R coding, there's one for communicating, there's one for emailing, and for different things. So we've got servers, we've got machines, we've got an idea that we have to train others, and they've got to learn, because this is a skill you've got to absolutely have, irrespective of what you're doing in the world. So I teach all of my data science using R, because the power of R is amazing. Those of you who are listening to me and have not used R before, please start learning R today, if not yesterday. All of the science that you do, all of the work that you do, any kind of data that you're looking at, whichever field of science or finance or medicine that you're working on, R will come to your rescue and help you with what you're doing. So during the pandemic, and this is something that has been appreciated, I think, by a lot of people, and it wasn't something I started out with, but it began as a lab meeting when the pandemic began. And during the lab meeting, I was trying to tell my own students and group members how to do different things, how to look at networks, how to improve, how to maybe cluster them, find communities, and so on. And then I realized that this is something that most people don't know, because we've not studied it in our courses. And so I decided to start doing something called Protocols from Home during the pandemic. And these are free courses that I just used to offer. And a lot of help came from Tigris, because my program managers in Tigris helped me a lot for setting up an event, for making it free, for registering people, letting them know when I'm doing these, how long they will be, and so on. Also from the Cambridge Bioinformatics department, I had a lot of help. And so these turned out to be very successful, tiny, I should say, tiny protocols from home, essentially. And I'm still continuing to do these. I can't do them very often because of my research and teaching responsibilities, both in India and at Cambridge. But if you're interested in joining me for any of these, you're more than welcome. Please connect with me on Twitter. My Twitter ID is JillianV. And whenever I do a course, I make it a point to post it out there. Even if I don't respond to your email or your phone call, I would post it out there on the social media, on my Twitter account, so that you would know when I'm doing the next course. And if you're interested, you're more than welcome to join us, okay? And the other thing that we did was in Tigris, I said repeatedly, but I can emphasize it one more time, we practice what we preach. So there is a big effort in Tigris where we want to enable women to do more than what they think they can. And especially during the pandemic, the digital gender divide we felt was hugely emphasized. And so what we decided to do was a course for women in AI, and it became hugely successful, especially for women who had never touched coding before. And we're going to run the second course in April, and I've now connected with the Delhi Effective Education Cluster, the DRIIV. You're welcome to take a look at us on the web. So if you're in Delhi and if you would like to get connected to the AI for children, AI for students, AI for schools effort, please get connected with me. We are currently looking for college teachers and school teachers in Delhi NCR region so that we can train them to train their students. There's just no way I can ever reach out to all the students that are there out there. But if we can train teachers, then you as a teacher would be able to connect to your students. And that's one way that I think we can really make a difference in spreading the idea of data science and AI all across India. So this is from our course, which was hugely successful. And on that positive and forward-looking note, I would like to end my talk and thank everyone who continues to enrich my experience as a plant scientist. I thank my students, my colleagues, friends, mentors, college members, old teachers, principal, the master at St. Edmund's College, Cambridge, where I'm a fellow, my teachers in India, my collaborators in both India and the UK, and most of all, my students, because all of the research, the awards, the work that we do, it actually is because our students are taking that effort in doing it. So my students in Cambridge, Ewan, Jamila, James, and Mark; in India, Situ, Abhishek, Vinita, Renu, Ravi, Sagar, thank you all. I especially thank Sastra for giving me the opportunity to talk about my work in a free-flowing manner. And of course, I have to thank my funding agencies in both the nations. And thank you all for listening. That's about it.
Thank you, ma'am. Thank you for the wonderful and inspiring lecture, inspiring in many ways, not just in terms of the research work that you do, but in terms of the teaching that you do and the leadership and the vision that you have. Extremely inspired. Thank you. Thank you for that wonderful lecture. May I request audience to post their questions on YouTube chat, whatever question that you have for Dr. Yadav, and we can raise it to her. I'm sure they're all spellbound, so we haven't had any questions so far. I think they were all spellbound. As we wait, a couple of questions from me, ma'am. One is slightly technical, so I'll just post it, but you can decide if you want to answer it. So you were talking about the temporal gene networks. It's curious how these little beings recognize this, that it is pre-dawn time and it is not yet dawn. Yes. So how do they actually recognize that?
It's a very good question. Thank you for asking that. So there's quite a few mechanisms. There's a lot that we don't know about plants, but what we know from the gene expression data is that they have a very strong circadian recognition. They know the circadian cycle very well. If you love gardening, and I think most of us do, you would know that plants flower just at the right time when they are supposed to. And that moment of flowering is called floral transition. And the plant knows when to flower. Similarly, the plant knows when to germinate. The plant knows when not to do it. And when there's light trigger, then it knows that you have to then start photosynthesizing. So all of this happens with, what should I say, signaling cascades or receptors that recognize recognize cues in the environment. So these could be external cues as well as internal cues, or a combination of both external and internal cues. There are hormones involved in this. There are signaling molecules involved in this. There are transporters involved in this. And there's a lot of connection with light and recognizing light. So that's one thing. But what is turning out now, and which is very interesting to note, is that in the past 5 years, there have been a few reports, not many, but there have been a few reports that there are some prion-like proteins also in plants, which have never been found in plants till now, but they have been reported to be having something to do with deciding flowering time. In, for example, the lumina dependence that was identified in 2016, and the ELF3 last year, which was found to have something to do with plants sensing and recognizing heat stress. So there are epigenetic signals also. That means histone methylation start to happen. So these are transgenerational changes that happen in the plant, as well as transient changes which happen just at the protein level. So many things are happening, and I think we need more plant scientists somehow out there to come and help us to decipher this better.
Okay, thank you. So we have one question from Dr. Uma. She asks, how accurate could be the predictions made using network analysis method?
Okay, this is a great question. Thank you, Uma, for asking. So the first thing that I want to say is, as I have often answered before in most of the courses that I take for artificial intelligence and machine learning, all of the computational and predictive methods that are being used today and that we can ever design in the future are knowledge-based approaches. So the moment I say it's a knowledge-based approach, it depends upon the knowledge that you have had about the system. So the lesser knowledge that you have, the worse is your prediction. The better knowledge you have, the better is your prediction. And therefore, it is extremely important to collect or collate as much information as you can. So whenever there's a new student working with me, or when you are starting on a new program, even when you're writing a grant, it's extremely important to do your, we are used to thinking review of literature is just another chapter, just write something. But that review of literature, that is the part that tells you what is known in the knowledge database prior to this. And that review of literature is your ticket to knowing what you can use for your predictive algorithm. More so currently, a lot of us in molecular biology and in genomics, we're using genome-wide data sets. So earlier, for example, we used to use these two hybrid to get to know what is interacting with what. And we used to do ChIP-seq studies to, even find out which transcription factor is targeting whom. But that was just one piece of information, or maybe a few. Right now, we are going for whole genome data sets. So you have ATAC-seq, you have MNase-seq, and you have large-scale RNA-seq data sets, which come with 6 replicates at least. And for each stage of the flowering, let's say, or in our case, we have the transcriptome data of the genome, all the 17,000 genes. We have their expression information for every half an hour of the day-night cycle of CLAMI. So we are looking at least 24 transcriptomes replicated 3 times, if not 6 times. And then we are taking into account this whole information. So there is so much available now to do a machine learning and unsupervised clustering on the gene expression data sets, for example. So we are now in a very good position, unlike 10 years ago, unlike 15 years ago. Today, we are in a position that if you've got a question about a process, I think you can very conveniently and very easily build a network, or a gene regulatory network, or a gene co-expression network, or a combination of these two, in order to find the regulators for that process. We have tried it for 3 different processes till now, and it's been hugely accurate. And of course, it depends upon what data you use. The larger the data set they use, the full genome data, if you can use, full transcriptome data sets, if you can use, the more they are, the better. Does that answer?
Okay.
Okay.
There's a allied question from Dr. Suma Mohan. So she says, great work in connecting data science and plant research. Thank you so much for the wonderful talk. It is really inspiring. She asks, what are the difficulties in validating your predictions? So okay. This is an allied question.
Thank you so much. Thank you very much. So the difficulties in validating, I would not put the question like this, because we are starting, unlike 10 years ago when I was doing my PhD, not 10, 20 years ago when I was doing my PhD, long back. So unlike those days, we were at that time looking at just the genome sequence, or we were just looking at the protein structure and trying to find some interface of protein interaction or the active. So my PhD is all about active site architecture and what happens with recognizing different parts of substrates by an enzyme. So at that time, we only had very limited data to work on. And so validation was an issue because I would predict that this protein interacts with B, and that's my prediction. But somebody had to prove, some experimental scientist had to prove that. Today, we've gone reverse because we already have the expression data, which means you've got experimental data to start with. So you're starting with experimental data, which means you already have the gene expression, but what you don't have, and which is seriously missing right now in plant biology, is people who can decipher that. So you've got the experimental data in front of you. You need a data scientist who can sit and find patterns there. I'm that guy, girl, who can do that, because most of our students, most of our friends, most of my colleagues, they're still looking at RNA-seq and doing DEG analysis. So all you do is differentially express genes and then you can publish that. But that's the tip of the iceberg. The entire gene expression is sitting under the DEG analysis. So let's get past the DEGs or DGEs and let's start looking at what, how across time, or how across stages, or how across the development, or how across mutations or stresses your complete transcriptome has expressed. And because you've got, this is much different from microarrays. That also 10 years ago, we were dependent on microarrays, which were also a proxy way of seeing whether there is expression or not. Today, you have exact transcript numbers and it's improving. And so I think the question of validating does not occur anymore because you've got validated data that's coming to you. You need to find the needle in the haystack. You need to be a good data scientist and you need to take some of my courses. Yeah, so that's the thing we need today. Thanks.
Thank you. So as we wait for more, if there are more questions, you can post it on the chat. I have one more question for you. This is on, the kind of work that you do is very exciting, but when does it actually get the, reach the masses? What is your vision for that?
Okay. So there are two ways of looking at things. One is that you can do blue sky research where the questions you're asking are to understand the fundamental properties of, for let's say the Rubisco study, let's say the efficiency of photosynthesis, the work we're doing in Tigris. So my part, out of 6 flagship projects, my part is to understand how biophysical CCM works. This is pure blue sky research because at best, I will find out how the alga are able to handle photosynthesis better. How will I translate that? The long-term idea in Tigris is that we should be able to translate the knowledge that we get from the unicellular green alga to higher plants and crops, right? So that is the leap of, that is the leap that you need to make. I cannot do this because I'm still working on the genes that are expressing. So your question becomes extremely pertinent, and the only way it can be solved, the only way the research can really be taken to the society out there is by connecting with others, as we've done in Tigris, by connecting with people who are working out there in the field, agricultural economists, also supply chain people, also people like our colleagues in Princeton and York who are doing the experiments. If we find a certain gene which we identify as very important for let's say biophysical CCM, can you then think about expressing it in higher plants? But the question is, higher plants may not have that gene altogether, because that's why the pyrenoid has evolved altogether in a different way. So there will be different kinds of questions coming in there. So this is blue sky research where you do something for getting a better understanding, but at the same time, in the same grant, you've got to be working with people on the ground at the grassroots level so that the research that they do. So for example, my colleague at ICRISAT in Tigris, they're working on millets, on trying to find out which are the most water use efficient millets. So Yana Kolova, Rajiv Gupta at ICRISAT, in the same project where I am looking at unicellular green alga, they're looking at across India from the last 50 years of data that is there with the agricultural department, which millets are used by which farmers, which are most effective for a certain geographical region. And we focused on certain states in the country. So once we find which are those millets, they're they're going to do they're going to transfer the data to me, and then I will do a GWAS with the least water use efficient and the most water use efficient millet. And so that would be a spectrum. From that spectrum, we would know which are the genes which are important for water use efficiency, and then we can immediately be in a position to translate that research to the field for those farmers, so that we already know which are the breeds which are the best, and we would know why they are. So that is the research that is translatable immediately, but both of them are in the same project. Does that answer your question?
Yeah. Thank you. So one of the reasons for my question is this involves a lot of networking and persistence over a longer period of time. And I think that is something that you have conveyed very beautifully to the audience, the young budding scientists, the students. And I think that is extremely important for them to know. Okay, Dr. Suma has responded saying, yes, exactly. So much hidden information in high throughput data. Thanks. So this was on the validation question. She has responded with the thanks. And then we have had Rashmi saying, wonderful talk, Dr. Gitanjali. Hemalada saying, very passionate and brilliant talk. And Dr. B.N. Rai saying, wonderful talk, learned a lot being unknown to the subject. So, yeah. So, if there are more questions we can take, or we can sort of bring it to a closure. Let's just wait for a few seconds, maybe. If there are questions, please post it.
No, I think it's extremely important to make your science, make people understand what it is. And I especially appreciate it when somebody who's not from the plant science background is able to understand even a little bit of what I've said. So I practiced my talk with my children yesterday, 8 and 10 years old, both of them. After a while, they said, Mama, just don't make it 1 hour long. It's too much. You try to finish it in 20 minutes. So I failed there today, but I think it's still worth it. And it's good to know that somebody who's not from a plant science background has been able to understand some bits of it. Thank you so much for that comment.
Yeah, so I'm sure your talk, as much as Prof. Ved left an indelible mark in you, I'm sure your talk must have left an indelible mark in many students who watch this talk and who will watch in the future as well. So with that, thank you again for giving us this wonderful and exciting talk, sharing your passion. I must honestly admit that your passion is very, very contagious. So thank you for that. And I'm hoping that the young scientists, the students, would take this forward, and we would have a great future. Thank you. Thank you very much, ma'am.
It's been a pleasure to be here today. Thank you so much.
Sir, we can go